Hello students, welcome you all in this video where we're going to complete our part C biology from SSLC model question paper 2 2025-26. Already we have completed part A physics and part B chemistry and I have uploaded the video. Please go through my channel, watch my videos, share my videos with your friends and also please consider subscribing my channel. So without wasting time, let's start with part C biology. So now we are going to start with part C, biology. And in this, the first section, that is question number 11, four alternatives are given for each of the following question or incomplete statements. Choose the correct alternative and write the complete answer along with its letter of alphabet. So it is a multiple choice question. We are going to start with this and this section carries three questions. Each question carries 1 mark. So total we are going to get 3 marks from this section. So we'll start with question number 26. Observe the figure below. The characteristic and function of the structure X are respectively. So here we have one figure. I hope you know this diagram. It is the diagram of rhizopus. So here they have labeled this X. You can see this. So this is the X and we have to tell the characteristic and function of this X. So options are option A, wall is absent and pollinates. Option B, it has a thick wall and grows into a new rhizopus. Option C, it has a thin wall and grows into a new rhizopus. And option D is, it has a thick wall and pollinates. So first we have to see this diagram and try to recall what is this. As I told, it is a diagram of rhizopus. And this is the example of spore formation. So it is an example of spore formation and it is nothing but the type of vegetative propagation, sorry, it is a type of asexual reproduction. So in asexual reproduction, spore formation is one of the method and for that we are using this example rhizopus. So here if you see these circles you can see these are called sporangia. These are called sporangia. And these sporangia, the bag-like or the sac-like structures, inside they have spores. So inside this sporangia, they have spores. And these, nothing but this is X, it is nothing but this spores. And these spores are the circular shaped structures. And this wall is very hard. This wall, which you can see, this wall is very hard and it is very thick. And because of this thick wall, when these spores get mixed with the water or the air, means when this sporangia get burst and spores comes outside, these all the spores will flow with the water, flow with the air, get stuck with the birds or the insect wings, and they travel a long distance. And till it gets a favorable condition, these spores will not get affected because it has a thick wall. And whenever it gets a favorable condition, moisture, it starts growing. So this thick wall helps to keep it or to prevent it from damage. So that is the main answer of this question. So therefore, if you see the answers options, it has a thick wall and grows into a new rhizopus. This is the correct answer, correct option for this question. So I hope you understood. So I told these circles are nothing but sporangia. These are the bag-like or the sac-like structure inside which there will be spores. And spores are the species or the organs which will grow to give a new rhizopus. But these spores will have a thick wall. Why they have thick wall? Because whenever the sporangia breaks and spores comes out, these spores will travel a long distance with the help of water or air or with the help of insects or the bird wings. And these in those long distance, when they travel a long distance, when they be in the environment, they will be safe because of the thick wall. And whenever they get a favorable condition, they starts growing to give a new rhizopus. So that's why the correct answer is option B. It has a thick wall and grows into a new rhizopus. So I wrote here answer. We have to write a letter of alphabet, that is B, and then write down the whole answer according to the question. So remember this, how to answer in the question only. It is given. We have to complete the answer along with its letter of alphabet. So remember, write B and then write the answer. This was the answer for 26. Now we are going to move with the next question on the next page. Now, question number 27, we will see the correct statement regarding bile juice among the following is. This juice is secreted by the. So, we have to write this bile juice is secreted by whom. So, option A, stomach and acidifies food. Option B, stomach and alkalizes food. Option C, liver and alkalizes food. And option D is pancreas and alkalizes food. So we have to select the correct option from these all. Now, as we know, once the food is entered into the mouth, since from the mouth, one by one juices will be secreted, and these juices have enzymes. These enzymes will help to digest the food step by step. So once it, as we all know, once it enters into the mouth, salivary amylase will be secreted, and which is secreted by salivary gland, and this amylase will help to digest the starch into the mouth. And once the food enters into the stomach has the gastric gland that will secrete the juice, and like this, the food, when it is entering into the intestine, in between the path there will be liver and pancreas. So this liver will secrete the juice which is called as bile juice. And this bile juice, once it's secreted by the liver, it get mixed into the food which is coming from the stomach, and it alkalize the food. Alkalize means what? There are three medium, as we know: acidic, alkaline and neutral. What do you mean by acidic? Acidic means the one which has pH less than 7. What do you mean by acidic? So I'll write it here. Acidic means pH should be less than 7. So I'll write it here, less than 7. And what is alkaline? Alkaline is nothing but basic. Alkaline, basic, both are same. So I'll write it here, basic. So remember, basic is nothing but alkaline. And here the pH will be more than 7. And one more medium is neutral, where the pH will be exactly 7 or is equals to 7. So here, once the liver, whatever bile juice is secreted by the liver, it mixes into the food coming from the stomach, the food will get alkalized, or the food will get a basic medium, which will help to digest that food which is coming from the stomach. So therefore, our correct option is option C, liver and alkalizes food. So remember here, write down answer. Letter of alphabet is C, liver and alkalizes food. So this is the answer for question number 27. Now we are going to move with question number 28. Question number 28 is, the correct statements about gene among the following is or are. So here we have some options here. One, it is a unit of DNA. So first is, it is a unit of DNA. Second, it is a source of information for proteins. Third, a single gene synthesizes several proteins. Fourth, it functions as an enzyme. So we have to choose from these all four. Either any one statement is correct or more than one statement is correct. That we have to see, and then we have to answer. So here we have the options. Option A, 1 only. Option 2, 1 and 2 only. Option C, 1, 3 and 4 only. And option D is 1, 2 and 3 only. So read the statements carefully and then choose the correct answer. Now if you see the statements, it is an unit of DNA. It is correct. What is the gene? Gene is nothing but it is a unit of DNA. What is DNA? DNA is a double helical structure like this. So in this double helical structure, let me draw the structure. So DNA has a double helical structure like this. So this, like this DNA looks, and there will be bases in this and sugars and phosphate group in this. So we know the DNA diagram you have seen. So in this DNA, there will be a particular portion like this. So these are the portions of DNA. So each portion is nothing but it is a unit, that is a gene. So gene, it is a unit of DNA. It is correct. Now see the second option. It is a source of information of protein. Correct. Every gene will be a source of information of protein. Means every gene will give the information about a particular protein, and therefore it is a source of information of protein. This is also correct. So I am telling once again, just understand what is gene. Gene, it is a unit of DNA. It is correct, and it is a source of information of protein. Means to synthesize the protein, this gene is the main source, which is the unit of DNA. So here first option also was correct, second option was also correct. Now we will see the third one. A single gene synthesizes several proteins. Actually, if we see, each gene will synthesize only one protein. It is the information of single protein. It will be, one gene will not synthesize many proteins. Remember, each gene is responsible for a single protein. It is a source of information of a single protein, not several proteins. Therefore, third option will be wrong. It functions as an enzyme. Gene will not function as enzyme. Gene is the source of protein, and proteins sometimes function as enzyme. Gene directly cannot function as enzyme. Therefore, this is also wrong. So if you see explanation, according to my explanation, according to NCERT book, third option and fourth option both are wrong. But first and second both are correct. It is a unit of DNA and it is a source of information of protein. And therefore, according to this, if you see option A has only one, but option B has 1 and 2 only. Therefore, our B option is the correct one. Option A has only one option, but according to our explanation, 2 is also correct. Therefore, we cannot mark A, we should mark option B. So write down answer. Option B, letter of alphabet is B, and the answer is 1 and 2 only. Now, whenever you write answers of this such type of questions, no need to write these statements again. Just write down whatever answers are there in the options. In options, they have given 1 and 2 only, so you should write only 1 and 2. No need to write one full statement, second full statement. Remember this. So this was the explanation of question number 28. After this, we are going to move with the next question. So now we will see question number 12, section number 12, that is answer the following question. And here again we have to answer 3 questions. Each carries 1 mark, so total marks from this section we are going to get 3. Question number 29, what is DNA replication? DNA replication means what? We have to explain. So answer, DNA replication is the biological process where cell creates an exact copy of its DNA before cell division, ensuring each new cell gets identical genetic material. So what is this DNA replication? Replica means preparing exactly same copy again. That is called as replication or replica. Exact copy is called as replica. So we know there are two types of cell division, mitosis and meiosis. Whenever this cell division takes place, one cell will divide to give another two cell. If we see mitosis, there will be one cell. This cell will be having nucleus at the center, then all the cell organelles, etc. Everything will be there. This cell, after maturation, it divides and gives two daughter cells like this. So this is mitosis I am telling. So here daughter cell will be having again nucleus and all the cell organelles exactly same as the parent cell. So if this is a parent cell, I'll write it here parent cell, then these two are daughter cells which we have got from the parent cell. So when parent cell divides to give two daughter cells, as I told, these two daughter cells will be having exactly same structure, same organelles. So to get this same structure inside the parent cell, whatever genetic material is there, all will divide or all will get duplicate or replicate into two. Nucleus will become two. All the cell organelles like mitochondria, Golgi apparatus, endoplasmic reticulum, everything will get double. And once it get double, half of the material will go into this cell and half of the material will go into this cell, and therefore daughter cells will look exactly same as parent cells, and this is nothing but the replication. So it is an important biological process. If this happens, then only we are going to get the same daughter cells as parent cell, and this will help to grow an animal as well as to transfer the characters from one generation to next generation. So therefore, the answer is DNA replication is the biological process where cell creates an exact copy of its DNA. Therefore, cell division ensuring each cell will get an identical genetic material. So whatever DNA it has inside the nucleus, exact copy of that DNA will form. Then one copy of a DNA will go in this cell, another copy of DNA will go in another cell. And with this process, this parent cell will ensure that both the daughter cell has got exact identical genetic material. So this was the answer for question number 29. Now we will see question number 30. Question 30 is, what is the relationship between amount of hemoglobin in the blood and the amount of ATP released in the human body? See the question, it's very simple. To see it is difficult, but it is very simple question. What is the relationship between amount of hemoglobin and amount of ATP released? So if the amount of hemoglobin is more, then what is the amount of ATP? It will be less or more. If the amount of hemoglobin is less, then the ATP amount will be more or less. This we have to answer. So as we know, hemoglobin, it is the material which carries oxygen in the blood. So everybody knows hemoglobin is present in blood, and this will help to carry the oxygen in the blood. Blood will move in all everywhere in all parts of the body, and when the blood is flowing in all the cells in all the parts of the body, it supplies oxygen to each and every part of the body. When it supplies the oxygen, every cell will take that oxygen, and that oxygen is used for respiration. Respiration means what? Burning of glucose. And whenever the glucose is burned, ATP is generated. What is ATP? It is nothing but energy. Adenosine triphosphate. That is nothing but energy. Energy is stored in the form of ATP. So remember, ATP is nothing but a form of energy. So whatever glucose food get burns into the cells, it produce energy which is stored in the form of ATP. So if more and more hemoglobin carries more and more oxygen, and then more and more ATP will be generated. If hemoglobin is less, then the amount of oxygen carried by this blood will be less. And if oxygen is less, ATP generated will be less. So I hope you're understanding. So that's why the answer is a higher amount of hemoglobin means more oxygen delivery to tissue, because more oxygen will be present into the hemoglobin, enabling greater respiration, greater aerobic. What is aerobic? The respiration which takes place in presence of oxygen. Respiration means burning of glucose. Burning of glucose takes place in more amount of oxygen. It is called aerobic. There is one more type of respiration which is called as anaerobic. Anaerobic means the burning of food in presence of less oxygen. That is called anaerobic. So here we are supplying more oxygen with the help of more hemoglobin. The speed or the greater amount of aerobic respiration takes place, and therefore greater amount of ATP formation occurs. And vice versa, if lower amount of hemoglobin means less oxygen delivered to tissue, and lower will be the aerobic respiration and less will be the ATP production. So these all are directly proportional. Remember, the amount of hemoglobin and the amount of ATP release is directly proportional. More is the hemoglobin, more is the ATP. Less is the hemoglobin, less will be the ATP formation. So this was the answer for question number 30. Now we are going to move with question number 31. What is the function of abscisic acid in the plants? So plants also produce many hormones like auxins, gibberellins, cytokinins, abscisic acid, etc. So here the question is, we have to tell the function of this abscisic acid. Answer: abscisic acid, the short form of this is ABA, mainly inhibits plant growth. Inhibit means what? Stops, prevents plant growth. Now you may think, why it will inhibit the plant growth? If it inhibits the plant growth, then plant will die. But this has a very important role. So see here, mainly inhibits the plant growth and promotes dormancy, acting as a key stress hormone that helps plants to survive in adverse conditions like drought, cold, high salinity, by closing stomata to conserve water, including seed dormancy. So what is the meaning of this whole answer is, as I told in Rhizopus, there will be sometimes adverse condition. Adverse conditions means in these conditions plant cannot grow or the seeds cannot grow. They will not get a favorable conditions to grow. So in such conditions, the seeds or the plant will stop its growth, and once favorable conditions appears, again it starts growing. So stopping growth, it will be helped with the help of this abscisic acid. So if there is a drought condition, if there is a highly cold condition, or there is a highly salinity formation, then it will help the plant to close the stomata. If it close the stomata means what happens? Water will be conserved, and this conserved water will help the plant to sustain in that diverse condition. And whenever there will be favorable condition, again the production of abscisic acid decreases and plant will become normal. As I gave the example of sporangia formation in rhizopus, the spores which are formed, these small circular spores, when they are flowing with the water or flowing with the air or flowing with the birds, insect or birds wing, they will not grow. They will remain constant. Means the wall will be so hard, so thick for the spores, they will not grow. They will remain as it is till they get the favorable condition. So why they remain as it is? Just because of this abscisic acid. So this will help to seed or the plants to remain as it is without till it get a favorable conditions, and that is the main role of this abscisic acid. So I hope you understood this question number 31. Now after this we are going to see the next question. So now we will see section number 13, answer the following question. Here we have to answer two questions, each carries 2 marks, so total we are going to get 4 marks. So question number 32, draw a diagram showing structure of nephron and label glomerulus. So here we have to draw the structure of nephron, which is the basic functional and basic unit of the kidney, which helps in excretion. Excretion means removing of unwanted waste substances from the blood. So these nephrons are the small basic units present in kidney. There will be millions of nephrons present in the kidney, and this nephron diagram we have to draw. So this is the diagram which I have taken from the NCERT book. You can see this label diagram which has all the labelings. Here we have to label only glomerulus, but whenever we practice, we should practice with all the labelings, because in this paper they have asked to label the glomerulus. Maybe in the final exam they may ask collecting duct, or they may ask Bowman's capsule. So remember, whenever we practice any diagram, we should practice with all the labelings. Now see the diagram. It consists of Bowman's capsule, then glomerulus, tubular part. It has two tubular parts. Then it has a U-shaped tube like this here, and it has this collecting duct. And then the remaining is those capillaries which we have to draw it here. This diagram, it looks very difficult by seeing this tubular part, but it is very easy to draw this diagram. So here, let me show you how to draw this nephron. So first what we should do, we should draw a Bowman's capsule like this. So see, like this we should draw a Bowman's capsule, and one capillary will go inside, which is called as renal artery, and the other one, that is renal vein, will come outside. So I will draw with the blue color like this, and inside this Bowman's capsule we should draw circles, and this is nothing but the glomerulus which is here. Now what we have to do, this is the pipe which is coming out from this Bowman's capsule. Here we have to take a turn, tubular turn. So if you see the diagram, it is a little bit complicated. What we should do, we should take a 3 turns like this, 1, 2 and third like this, and complete this all. See here, so simple. Now this we have to draw a U-shaped tube like this, and then after drawing this U-shaped tube, again we have to take a coil. So take some 3 coils like this. See here, 3 turns I have done, 1, 2, 3, and then collect this turn to this collecting duct, and like this we can complete the diagram. Here the turns which you take, it is depend on you. No need that it should be same as NCERT book. After doing this, now we have to take the capillaries, and capillary how we have to do, see here. So the pipe which is coming out, let me keep this red itself. The coming out, it comes here and split it into two like this. One will go inside these all the pipes, and another one, it goes like this and comes on the other side of this tube. And here we have to do a network like structure. Network means connect here like this. See, like this we have to complete, and this is the diagram of nephron, which is very simple to draw. Now in this diagram we have to label glomerulus, and the glomerulus is here, glomerulus. But whenever we practice, you should practice all the labelings of every diagram. So this was the answer for question number 32. Now after this, we will see question number 33, which has again A and B part. Question number 33, the A question is, name the prominent parts of reflexing arc. Reflexing arc, it is the path in which the stimulus will travel. The stimulus, sudden stimulus which is given, it is travel, that is called as reflexing arc. So see here no need to draw the diagram, just we have to tell the prominent parts. So we have a receptor in the muscles which receives the stimulus or detect the stimulus. Stimulus means what? These are the external signals which will be received by the receptors. It may be a heat or a cold or a touch, smell, etc. So once these receptors receives or detect the stimulus, that stimulus will be carried by the sensory neuron to the CNS, central nervous system, where there will be spinal cord, which will process on that signal or the stimulus which is received by the receptor. And after processing, after working on that signal, the back message will be given to the motor neuron, and this motor neuron will carry that back message, or it is called as impulse, from the central nervous system and give it to the effectors. Effectors are present in muscles where, which will show or respond to that stimulus. Like when we touch a hot plate, that heat is nothing but the stimulus. that a message is carried by the sensory neuron. It is given to the spinal cord, which will work on that message signal, and to remove the hand, that back message will be carried by the motor neuron and it will be given to the effectors. And the muscles which are there in the effectors will goes aside, means we will remove our hand from that hot plate. And that is nothing but our sudden action or the reflex arc. So this is the answer for question number 33A. Question number B is, which hormone is responsible for sexual maturation in humans? So human means we have to tell the hormones which are responsible for sexual maturation in both male and female. So first we will see testosterone, which is also called as androgen, which is present in males. And whenever this hormone will be secreted, there will be a secondary sexual character which are grown, like growth of the facial hair or body hairs, deeper voice, muscles will increase, or there will be sperm production. So these all the secondary characters which we can see in males, and the hormone is testosterone or also called androgen. So here the next is estrogen and progesterone. So here estrogen, it is the hormone which is responsible for sexual maturation in females, and progesterone, it is the hormone which helps in a hormonal balancing in case of pregnancy rule. So due to this estrogen, which is produced in females, we can see the secondary sexual characters like breast development, menstruation, etc. And progesterone will help at the time of pregnancy. So here they have asked only hormones. So remember to write testosterone or androgens, estrogen, progesterone. So these are the two hormones. This is the extra information which I have given. It will help you to answer some other questions. So remember these all the information. So this was the answer for B question or B part for question number 33. After this, we will see the next question. So now we will see the next section, that is section number 14. Answer the following question. And here we have to answer 3 questions. Each question carries 3 marks. So total we are going to get 9 marks from this section. Question number 34 we will read. Draw the diagram showing the structure of human brain and label the following parts. So here we have to label cerebrum and pons, and we have to draw the human brain diagram. So you can see this is the diagram of human brain. It is from again NCERT book, and here we have to label cerebrum and pons. So here there is the cerebrum, this we have to label, and pons is here, so this we have to label. So these are the 2 labelings which we have to do for this question. And remember, drawing this brain is little bit difficult, but if you remember some of the tricks, then it will be easy. I will be doing a separate video to show that how to draw this brain easily. And remember, whenever you draw these 3 layers outside, and then you draw this middle part, and after drawing these 2 parts, then you draw these lines. Drawing these lines is very easy. Then remember, as I'll be telling always, whenever we practice, you practice these all the labelings which are there in this diagram. Like first is forebrain, then here there is a hypothalamus, then there is a pituitary gland. So this pituitary gland, hypothalamus also important. Sometimes that also will be asked to label. Then there is a hindbrain which consists of 3 parts, that is pons, medulla and cerebellum. Remember, cerebellum is different and cerebrum is different. So while reading, read the question carefully and answer. In this question, they have asked cerebrum. So cerebrum is here. Then here there is a spinal cord. This center one is called midbrain, and this is cranium, that is skull. So these are the labelings of human brain. So what is asked to label in this question? So here we have to label cerebrum. This is the one, and the second one is pons, that is here. So these are the 2 labelings which we have to do in this question. Now we'll move with the next question. So now we will see the next question, that is question number 35. Red flowering tall plant, that is capital T, capital T, capital R, capital R. So here we have 2 characters, that is 2 traits. One is color of the flower and second one is height, that is tall. Is hybridized with white flowering dwarf pea plant. So another color is white for the flower and height is dwarf, that is small t, small r, small r. So remember, red and tall is the dominant character, white and dwarf is the recessive character. So now show the result of F2 generation with the help of a checkerboard. Write the ratio of varieties of plants. So here we have to show the result of F2 generation. But before going to F2 generation, first we have to solve for F1 generation and then we will do the F2 generation. Then it will be easy for us. So we will solve it step by step. So as we have here two characters, that is red color of the flower and height of the flower. that is red and white, and tall and dark. So it is a dihybrid cross. So I will write here first dihybrid cross. So it is a dihybrid cross. So first we will start with F1 generation, and for this we will write here. So what is our plants? Red flower and height is tall. So I'll write tall plant. So what are the genes denotion for denoted for this? So red is capital R, and tall is capital T. So we will denote it like this. So we will write it here. Now same way we will write for another plant, that is white flower, and what is the height? It is a dwarf. So write down dwarf plant. And how we will denote this one? So we will write it as small t, and the another thing is, sorry, we will make the correction. As here first we have color, so we will write small r, and then we will write small t. So this is the main plant, and now we will start doing the F1 generation. So write down capital R, capital T. So capital R twice, so we'll write only once. Here the genes will be small r and small t. Now when we cross these two, now see these two plants are different plant. So therefore this is called cross pollination. So this is cross pollination. So I'll write it here, cross pollination, because this plant is different, this plant is different. Now what we are going to get? Capital R, capital T, and small t. And what is this? This is the result of F1 generation. I'll write it here, F1 generation. So F1 generation, and this is the genotype. Then what is the phenotype? How the flowers will be? So see the color, it is capital R and small r. So capital R is dominant. So still the flowers, whatever we are going to get in F1 generation, all will be red flower. And if you see the height, it is capital T. Capital T is there, means it is dominant. So again, here the height will be tall. So tall plant. And this is the phenotype of F1 generation. So here F1 generation complete. And now we will go for F2 generation. So here I'll write now, we'll draw one line, and then we will write here F2 generation. Now for F2 generation, what we are going to do? We are going to take the plant from F1 generation and self-pollinate it. So write down here capital R, sorry, small r, capital T, and small t. And this we are going to self-pollinate with the same plant. So I'll write same plant once again like this. Now see, now this we have to write the genes. Now how to write the genes for this? So very easy. So here I will give you one hint. Capital R combines with capital T, capital R combines with small t. Then small r combines with capital T, small r combines with small t. And same way we have to do this side also. So like this, like this, like this, and like this. So what are the genes you are going to get? Capital R, capital T, then capital R, small r, capital T, small t. And here also same thing I'm going to get. Capital R, capital T, capital R, small r, capital T, small t. So these are the genes which you're going to get from these plants. So we are going to get 4. Now we have to cross this, and if you cross it with the help of lines like this earlier, it will be too confused. So therefore for this we will use a checkerboard. So let me draw the checkerboard here on the other side. So F2 generation we are going to do in this. So I'll write it here, F2 generation, and with the help of checkerboard we are doing, so by checkerboard. So write down here checkerboard, and now write down the genes. So for this plant, we will write it on one side. I'll write it here. That is capital R, capital T, capital R, small r, capital T, and small r, small t. Then the other plant, this plant, we will write it on the vertical side. So here I'll write capital R, capital T, and then capital R, small r, capital T, small r, small t. Now these both are same plant, therefore in these two plants whatever crossing we are doing, it is self pollination. So we will write it here, self pollination. Now we will cross and write down all the result what we are going to get. So see here, capital R, capital R, capital T and capital T. How we got this? By combining these two. Then capital R, capital R, capital T, small t. Then capital R, small r, capital T. Then capital R, small r, capital T, small t. Then here also same thing, capital R, capital T, small t, capital R, small t, capital R, capital T, small t, then capital R, small t, capital R, capital T, capital R, capital T, and this is small r, capital T, small r, capital T, and small t. Last one, capital R, capital T, and small t, capital R, small t, and then small r, capital T, capital small t, and small t. So this is red tall. This is red tall. This is red tall. Red tall. The small t means it is dog. So we will write it here, dog. Then again complete this by saying dominant and recessive gene. So we have completed all this checkerboard. Now we will count how many red tall, how many red dwarf, how many white tall, and how many white dwarf. So this is red tall 1, 2, 3, 4, 5, 6, 7, 8, 9. So we will write here red tall. We have 9. Let's write it right. Then the next, now we will see red dot. So calculate first how many red dots. So here there is a red dot 1, here there is a 2, and this is 3. So write down 3. So write down here 3. Now we will see the next one. That is, the next one is white tall. So I'll write it here, white tall. How many white tall we have? So check how many. 1, 2, and 3. So white tall 1 and last white dog. White tall is 3. These 3 and white dog is only 1, which is here. So write it 3 here, and the last as I told, white dog, it is 1. So the ratio is 9 is to 3 is to 3 is to 1. So this is, so it is a genotypic ratio. Sorry, it is a phenotypic ratio. Phenotypic ratio of F2 generation. So write it completely. This is the answer for question number 35. So now we will see next question, that is OR question from question number 35. So this is again from heredity chapter. Yellow seed producing pea plant is hybridized with green seed producing pea plant. So here yellow seeds are denoted by capital Y. So it is a dominant one, and green seeds, it is denoted by small y. So it is a recessive one. Now see here we have only color of the seed. So we have only one character, only one trait. So it is a monohybrid cross. The earlier question was the one more option in the question number 35. It was dihybrid. Now this is monohybrid. I'll read the next. Show the result of F2 generation with the help of checkerboard. Write the phenotypic and genotypic ratios of F2 generation. See, it is very simple. As it is a monohybrid, it will be very simple to draw this checkerboard. So we will start with the answer. So in the question, they have asked F2 generation, but we have to start with F1 generation. So I will start. So I'll write here first we have yellow seeds. So I'll write yellow seeds. So this plant is having yellow seeds, and how it is denoted? Capital Y and capital Y. So it is dominant. Then we have another plant which is giving green seeds. So I'll write it here, green seeds, and green seeds are denoted by small y, so it is recessive. Now what are the genes from this? So we will write gene capital Y, because both y are same, so we will take one capital Y, here we will take one small y. Now these two plants we have to pollinate. Now these two plants are two different, so therefore this is cross pollination. So we will write it here, it is a cross pollination, because the plant which is giving yellow seed, it is different, and the plant which is giving green seed, it is different. So two different plants we are going to cross. So it is cross pollination. And when we cross, we will get capital Y and small y. So this is the F1 generation. So I'll write it here, F1 generation. And in this, we can see whatever plants we are going to get in this F1 generation, all plants will have yellow seeds. All plants will have yellow seeds. Why all plants will have yellow seeds? Because if you see this notation for this F1 generation, the genotype, it is capital Y and small y. If it is having one capital alphabet, that is the dominant, and that will be the dominant gene or dominant character will be seen. Therefore, capital Y means yellow. So all the seeds which we are going to get in F1 generation, all will be of yellow color. So this was F1 generation. Now we will see F2 generation. So here I'll draw one line and then I'll write F2 generation. So draw one straight line. I will write here F2 generation. And in this F2 generation, I am going to cross the plants which we have got in F1 generation. So I will take here what plant we have got here, the plant which is having yellow seeds. Yellow seeds. And what is the notation? How we will denote it? Capital Y and small y. You can see here in the F1 generation, we have got capital Y and small y. This is yellow seed, and this first also yellow seed, but this is homozygous. This was both are dominant, but here in this F2 generation, one capital Y and one we have small y. Same plant we are going to cross, in the same plant we are going to cross it. Therefore, this is a self-pollination. So I will write here again one more, that is yellow seeds. We are going to do the pollination in the same plant, and therefore it is a self-pollination. Now what are the genes we will write? Here both are different, so we will write separately capital Y, small y, and here also same thing. We will write capital Y and this is small y. So these are the genes. Now these all we have to cross. So when we cross these two, so which type of pollination it will be? It will be of self-pollination. Remember, because in the same plant we are going to pollinate. So it is self-pollination. You can see in the F1 generation, these both the plants are different. Here both the plants are same. So when we cross, see there, this Y, if it combines with this Y, we will get capital Y and capital Y. Now same y, if we combine with this small y, we will get capital Y, small y. Now again this and this, if we cross, we will get capital Y, small y. And last, this y and this y, if you cross, we will get small y and small y. So like this we will get F2 generation. But this will be little bit of difficult to understand. So we will make it easy with the help of checkerboard. Also you can see the question, they have asked to use the checkerboard. So with the help of checkerboard, we will do it at the side. So here this is our checkerboard. So I'll write it here, checkerboard. Now write down the genes what we have. We have one plant with capital Y and small y. Other plant also with capital Y and small y. Now if you combine this Y and Y, we will get capital Y, capital Y. Second, this Y and this Y, we will get capital Y and small y. Then this small y and this capital Y, again we will get capital Y, small y. This Y and this small y will get small y and small y. So if you compare this with the earlier which we have done here, all are same. Capital Y, capital Y. See here, capital Y, capital Y. Capital Y, small y, we have 2. So here also capital Y, small y, we have 2. And small y is 1. So small y is 1. So this is the F2 generation with the help of checkerboard. And this is a simple, normal method, regular method. So now we will see. So in the first one, capital Y, both are dominant. So here whatever seeds we are going to get, all will have yellow seeds. See here, all will have yellow seeds. Here also we have capital Y, so here also whatever seeds will be there, all will be of yellow. This also will be yellow because there is one capital Y. So I'll write yellow seeds. But the last one, if you see, both are small y, small y. So it is recessive. So here, whatever seeds we are going to get, all will be of green color. So I'll write it here, green seeds. So this is called as checkerboard. So whatever these all capital Y, capital Y, capital Y, small y, these all are genotype. And this yellow seeds, what I have wrote, it is showing phenotype. So we will write it here. So genotypic ratio, because it is asked in the question. So genotypic ratio. So if you see the ratio, it is, we have capital Y 1, then we have capital Y, small y 2, and we have small y, how much? 1. So therefore, the ratio will be 1 is to 2 is to 1. This is genotypic, where we can see the genes. According to the genotype, we have done the ratio. Now we will do phenotypic ratio. Here we have to see the physical character, the color of the seeds, and according to that, we have to write the ratio. So if you see now, so yellow seeds, how many we have? We have 3. This is also yellow seed, this is also yellow seed, this is also yellow seed. So I'll write it here, yellow seeds. How many we are getting like this plant? We are getting 3. So I'll write it here 3. So 3 is 2. We are getting green seed also. So I'll write it here green seed. And green seeds plants, how many we are getting? We are getting only 1. So I'll write it here 1. So this is 3 is to 1. So I hope you understood here. So here genotypic ratio is 1 is to 2 is to 1, and phenotypic ratio is 3 is to 1. So these are the two ratios. So according to question, our answer completed. So F2 generation, this is the genotypic ratio and phenotypic ratio. Now we'll move to the next question. So now the next question, that is question number 36, which is divided into 3 parts again, A, B and C. We will start with the A. What are biodegradable substances? So see the question, what are biodegradable substances? Degrade means decompose. Bio means living. So we have to tell the substances which are decomposed with the help of biological organism. The substances which are decomposed with the help of biodegradable or biological organism, which is called as biodegradable substances. So these are natural material like food scraps, paper and leaves that can be broken down by the microorganisms like bacteria, fungi into simpler harmless substances enriching the soil. So here what we are going to do, we have to tell which are the substances, which substances are called as biodegradable substances. So as I told, if you see food scrap, the waste, food waste or paper or leaves, dry leaves, these all if you throw in a soil, on those substances, microorganisms will grow, like bacteria, fungi will grow. If you see, if you keep bread for more days as it is without touching it, if you forgot to see the bread or the roti which will be there in the tiffin, then after some days you may see there will be some black or the yellow color cotton like substance will grow on that bread or the chapati or roti. So what is that? It is nothing but some kind of fungus will grow on those all the materials. Especially on bread, you maybe have seen some black material will form after 4 5 days. So that is nothing but the microorganisms which grows on the bread, and those all microorganisms will eat those all the bread or the roti or this food waste, and it will convert that into a simpler harmless substances, and again it will get mixed into the soil. Why it is saying enrich the soil? Because if you throw any natural waste like dry leaves, if you see whatever the dry leaves will fall down from the trees, those all the dry leaves will fall on the land or the on the earth in the soil. Those dry leaves on the earth in the soil will be decomposed with the help of these microorganisms, and again it will get mixed into the soil. So it will enrich the soil. That is the meaning. That is why the enrich the soil word is used here. So the substances which can be decomposed with the help of microorganisms. Microorganisms are living. Therefore, it is called bio. So that is the answer for question number A. Now we will see B. Which synthetic chemical is responsible for the depletion of the ozone layer? So what is ozone layer? Ozone is nothing but O3 molecule. O3 molecule which is called as ozone layer, and it is the layer present beside the earth in the atmosphere if this is the Earth, then in the atmosphere, at some far distance from the Earth, there is one layer present which is called as ozone layer. Ozone, it is nothing but the formula is O3. So this is called as ozone layer, and this ozone layer will help the Earth. It prevent the harmful radiations which are coming from the sun. So if the sun is liberating, as we know, electromagnetic waves are coming from the sun. These waves will have their ultraviolet waves, UV rays. Those are very much dangerous. Those waves should not come in contact on the earth or with the living beings. And those all the rays, UV rays which are coming from the sun, these UV rays will be trapped in this ozone layer. And because of those UV rays trapped in this ozone layer, the living uh, organisms which are there on the earth will not get damaged by these rays. But because of some chemicals, this ozone layer is decreasing, or it is called as depletion. It is decreasing. So that chemical we have to tell. So answer is the synthetic chemical like chlorofluorocarbons. It is very important you have to remember this. The chemical like chlorofluorocarbon, that is shown by CFCs, capital CFC, then small s, which are used as refrigerant and in fire extinguisher. So this is one of the chemical which is used in refrigerator as refrigerants, also used in fire extinguishers. These gases or these chemicals are responsible for decreasing the ozone layer. So due to these chemicals, ozone layer is decreasing. So nowadays these chemicals have been banned. Some other chemicals have been replaced by the CFCs. So these chemicals are decreasing the ozone layer, and because of this, these UV rays, instead of preventing in this ozone layer, they are trying to come on the earth. And if these comes on the earth, if they come in contact with the living beings like skin, especially in human being, if it comes in contact with the skin, they are causing skin cancer. So that is the main problem which is caused due to this uh, depletion of ozone layer. So this was the answer for question B. So remember here they have asked the chemical. So chemical is CFCs, that is chlorofluorocarbons. Now we'll see the next, that is C. Write two examples of decomposers. So we have to give the example of decomposer. What is decomposers? These are the organisms which will decompose. So there will be, if you see the tropical levels, if you see the tropical levels, first there will be producers, then there will be primary consumers, secondary consumers, tertiary consumers, and then there will be decomposers. So once the animal or the plant dies, it has to decompose. It has to broken into simple parts so that it will get mixed into the soil to enrich the soil, and again the cycle will continue. The living being cycle will continue. So we have to tell which are those decomposers. These are the main important animals which will help to decompose or to continue the cycle in the nature. So two common examples of decomposers are bacteria and fungi. So as I told, fungi means like mushrooms. Mushrooms also grow on the dead animals or the dead living beings. So here see, bacteria and fungi are the main example which break down dead organic material and returns nutrients to the soil. Whenever any animal dies or some waste is thrown into the soil, especially biological waste, those all will be decomposed. Why? Because fungi and bacteria like microorganisms will grow on that. They will eat those all the waste, means they convert those big materials into smaller materials and mix into the soil. There are some other organisms like earthworm, insects, and millipedes that also consumes and process dead matter. So leaving this bacteria and fungi, there are some other organisms also. So you can write earthworms, insects, and millipedes. So mainly you can write these two in the answer, bacteria and fungi. So this is the answer for question number C. So here we have completed question number 36. Now we'll move to the next question. I hope you are understanding all the questions, all the answers which I am explaining. Make a notebook, write these all the answers in your notebook so that whenever there will be exam, it will be help for you people for doing the revisions. And share my videos with your friends, and please consider subscribing my channel. Now we'll move to the next question. So now we will see the next section, that is section number 15. Answer the following question. Here we have to answer only one question, but that question carries 4 marks. So from this section we are going to get 4 marks. So question number 37, which is divided into A and B. So we will see the A option. What are the functions of aorta and pulmonary veins? So read the question carefully. It is vein, it is not artery. So we have pulmonary artery and pulmonary vein. So pulmonary artery is different, vein is different. So read it properly. So we have to tell the function of aorta and pulmonary artery. So what is the answer for this? The aorta's function is to carry oxygen-rich blood. It is nothing but oxygenated blood, the blood which is having oxygen, from heart's left ventricle to the rest of the body, delivering oxygen and nutrients. So remember this, aorta will help to carry oxygenated blood from the heart to the rest of the body, all parts of the body, to supply this oxygen and nutrients. And what about pulmonary vein? It carry oxygen-rich blood, that is oxygenated blood, from the lungs back to the heart. So, as we all know, blood will be purified in the lungs. Once the blood is purified in the lungs, from lungs the blood will come to the heart in the left atrium, and that will be carried by this pulmonary veins. And once it comes into the left atrium, from here it goes to left ventricle, and from left ventricle aorta will carry this oxygenated blood to all parts of the body. We can understand it still in a clear way. So see here, I will draw one simple diagram, a simple box. So our heart is having four sections. So imagine this is our heart, which is having four sections here. And this is a left atrium. So I'll write LA. This is left ventricle. This is right atrium. And this is right ventricle. Now see what happens. Here, imagine this is our lungs. So here somewhere we have our lungs. So this is lungs. Now, from lungs, here the blood will be purified. So I'll write blood is purified. So we are going to purify the blood in the lungs. Once the blood is purified, that purified blood or oxygenated blood comes to left atrium, and this will be carried by who? This is pulmonary vein. So I'll write here pulmonary vein. So pulmonary veins carry oxygenated blood from these lungs to the heart. So this is our heart here. So here I'll write this is heart. Now see. So this is the second answer, that is blood from lungs, oxygenated blood from lungs to heart will be carried by pulmonary veins. Now once the oxygenated blood comes to left atrium, it goes to left ventricle, and from left ventricle it will be forced and passed to this aorta. So this is our aorta, and this aorta will take this blood to all parts of the body. So I'll write here all parts of body. So see, so aorta carries oxygenated blood from left ventricle to all parts of the body, and pulmonary vein carries oxygenated blood from lungs to left atrium. So once it goes to all parts of the body, here in the cells again exchange of gases takes place and the blood becomes deoxygenated, and this deoxygenated blood again comes back to the right atrium. It will be forced to right ventricle, and from here pulmonary artery will carry this blood again back to lungs. Here it will be purified and the process repeats once again. So this is the role of heart. I hope you understood this answer. That is question number 37A. Now we will move with B. So here B question, state the events that occur during the photosynthesis in plants. So plants will do photosynthesis with the help of chlorophyll. So what are the steps will occur in the photosynthesis, that we have to answer. So in NCERT book they have given clearly all the steps, same steps I have taken here to explain you people. The events that occur during the photosynthesis in plants are, first one, absorption of light by chlorophyll. So first chlorophyll will absorb the light or the light energy. Light is nothing but a form of energy. Therefore it is here energy word is used. So first green color chlorophyll substance will absorb the light. Once that light is absorbed, that light energy, conversion of light energy into chemical energy and splitting of water molecule into hydrogen and oxygen. So once the light energy comes into the chlorophyll, that energy is used to split this water molecule to form hydrogen and oxygen, means we are preparing the chemicals. So means what, we are converting light energy into chemical energy. And now reduction of carbon dioxide to carbohydrates. Now what is reduction? Reduction means addition of hydrogen. So this hydrogen which is formed, that will be added to the CO2 which is there in the plant and forms carbohydrates. And whatever oxygen is formed, it is a byproduct. That's why plants will produce the oxygen. We will say plant produce oxygen, and that is why here we will get the oxygen. So these are the steps which occurs during photosynthesis. Let me explain with the help of diagram. So here I will draw one leaf. So this is the leaf. It will be good to use this leaf. So I'm drawing this leaf to explain this answer. So see, this is the leaf. Now leaf will have the green color substance everywhere, and that is nothing but chlorophyll. Now here somewhere there is a sun, and from this sun we are getting the light energy. Once this light energy comes and it falls on the plant, I'll write it here. This is our light. Once this light energy comes and falls on the plant, it will be trapped by this chlorophyll which is present in the leaf, and that light energy will be used to break this water. Water is already there in the plant, so it breaks this water and forms hydrogen and oxygen. So what is this hydrogen oxygen? This is nothing but chemicals. So I'll write it here. These are chemicals. So how we got these chemicals? From light energy we have converted into chemical energy. Once this hydrogen oxygen is formed, already there is a CO2 in the plant. So this CO2, you can see in the CO2 there is carbon and oxygen. So whatever hydrogen is formed, it will be added to the CO2. Addition of hydrogen means what? It is reduction. So here reduction takes place. So reduction occurs, hydrogen is added, and after adding hydrogen, the CO2 becomes C6 H12O6, nothing but glucose. It will form here carbohydrate. I am writing glucose. It is nothing but it is called as carbohydrate. So food is prepared. So I'll write it carbohydrates are formed. So this is photosynthesis. We got the food. And what about this oxygen? This is the oxygen which will be produced by the plant as a byproduct. And this oxygen we all living organisms, animals are using for respiration. So these are the steps which occur during photosynthesis. So I hope you understood these all the steps. These all the steps are also there in the NCERT book. From there only I have taken this to explain you people. Now we are going to move with the next question. So the next question, this is the OR question in this. So OR question again divided into A and B. First we will see A. In the first stage of cellular respiration, the glucose molecule is break down into which molecule in the cytoplasm? This, how is this broken molecule breaks down in the muscles? So see here, this is the question of respiration. So in respiration, glucose will be broken down into CO2 and H2O. This is the main process of respiration. So when this glucose is broken down, this takes place in two steps. In the first stage, this respiration takes place in cytoplasm, and after that stage, the second step takes place. Second step is explained in three places. That is, first we are going to see in yeast, then we will see in muscles, and then we can see in mitochondria. So in these all the things, in these all cases, the first stage will be same. That is the respiration which takes place in cytoplasm, but only in second stage there will be three cases. So here they have asked, once the glucose is broken down in the cytoplasm in the first stage, what we are getting, which molecule we are getting, and after this we have to explain second stage, but we have to explain the case where respiration takes place in muscle cells. We no need to explain about the yeast and the mitochondria. So we will start the answer. So number one, the first step is the breakdown of glucose, a 6 carbon molecule, into 3 carbon molecule, which is called as pyruvate. So once the glucose, once the oxygen is reached to the cell from the blood, that oxygen will be taken by this glucose and converted into 6 carbon molecule, which is called as pyruvate. This whole process takes place in cytoplasm. So you can see here, these all the notes I have taken from NCERT book. You may see this reaction. Glucose, which is 6 carbon molecule, that is C6H12O6, in cytoplasm, it is broken down into pyruvate, that is 3 carbon molecule, and with this it will release some amount of energy. But it will not release more energy, because still this pyruvate can be broken down into simpler substances. Then only we can get the more amount of energy. So here we will get small amount of energy. Now, when this pyruvate, there are three cases as I told. This pyruvate can be broken down in the yeast, that also we can study. Pyruvate breaking down into the mitochondria, that also we can study, and even this pyruvate breaking down into the muscle cell, that also we can study. In this question they have asked about muscle cell, so we will explain, we will see only the case where respiration takes place in muscle cells. So in muscle cell, when there is a lack of oxygen, see, now this pyruvate enters into the muscle cell where there is a lack of oxygen. This pyruvate converted into lactic acid. So this 3 carbon compound, which is pyruvate, will not be broken completely because there is a lack of oxygen, less amount of oxygen. and that pyruvate will convert into lactic acid, which is again a 3-carbon molecule, and it releases some amount of energy. So whatever energy released in this process, that is also very less, because this lactic acid is not the last compound. This can be again broken down into simple one. But because there is a lack of oxygen in the muscle cell, pyruvate will give this lactic acid and some amount of energy. This production of lactic acid in our muscle during the sudden activity causes cramps. So when this reaction takes place in muscle, this lactic acid is produced and this will be stored in the muscles. And when when the amount of this lactic acid increases, exceeds, it causes cramps in the muscles. That's why you maybe have seen when we run or when we do jogging or when we do some amount of work, continuous work or lifting some weights, etc., then you may feel some type of cramps in the muscle, and these all just because of this process. Because whenever we do some kind of this work where the muscles are used, more amount of energy is required, and in muscles, because of lack of oxygen, it will not create more amount of energy, and that's why the breathing process will be fast when we do the work. So here remember, the first question answer is pyruvate, which is a 3-carbon compound. And in the second step, how is this molecule is broken down into muscle? So this pyruvate molecule in muscles broken down to give lactic acid and energy. And remember, if the question asked why the cramps will be caused in the muscles, that is just because of deposition of this lactic acid. So this is the answer for this A. Now we will see B. Write any two differences between xylem and phloem. So xylem and phloem are the complex tissue which are present in the plant. So we have to tell the differences. So here, whenever the differences or such type of questions are asked, try to write those answers in the box, in the columns, so that it will be easy to compare the differences. So one side I wrote xylem, the other side I wrote phloem. So xylem transports water and minerals. So exactly in the same way, but in the opposite, we have to answer for the phloem. Should not mix all the points. Like here you are telling the work, the function of the xylem, and this side you are telling the structure of the phloem. Should not do like this. If you are writing the function this side, then you should write functions of phloem this side. That is called as difference. So first point is xylem transports water and minerals. So remember, what is the role of xylem? Once the root absorb the water which is having minerals, these water minerals are transported to all parts of the body with the help of all parts of the plant with the help of xylem. Now, what about phloem? Phloem transports food, that is nothing but sugar. So, whatever food is prepared in the leaves with the help of photosynthesis, that food will be transferred to all parts of the plant, because whenever the plant grows, all parts of the plant will grow. And for all parts, food is required, and that transport will be done by phloem. Second point, xylem, that is unidirectional, only from roots to up means plant, top of the plant. So we know that roots will absorb the water. Roots are always down and the plant will be up. So here, once the water is absorbed by the roots, it will be carried to all parts of the plant which are upside. So it is unidirectional. But phloem is bidirectional to all parts of the plant. So as we know, so let me explain you with the help of diagram. So see here, if these are the roots of the plants and this is the plant. So these are the twigs, parts, these are your leaves, there will be flowers, fruits, etc. etc. So see here, in terms of xylem, these roots will absorb the water with the minerals, and the xylem will take this water with the minerals to the plant and it supplies to all parts of the plant, and it will be unidirectional. It moves only upside, upward. But in case of phloem, leaves will produce the food. Now see, imagine this leaf, it is producing the food. So whatever food is produced in these leaves, it has to be carried upwards to all parts of the body, all parts of the plant, as well as it should be carried downwards to all the roots and remaining parts of the plant which are present downwards. So phloem will work in bidirectional, upwards as well as downwards. But xylem will work only upwards in unidirectional. So that is the meaning of this point. The third point, xylem composed of mainly dead cells. So whatever cells are present in the xylem consists of vessels, tracheids, xylem parenchyma, and xylem phloem. So here these vessels and tracheids are mainly dead cells which are helping for support. But in case of phloem, it consists of sieve tubes and companion cells, and mainly these cells are living cells. So these are the main important differences which you have to write for this question. They have asked two differences, but here I have given three differences. Remember at least three differences. It will be easy. If you forgot any one difference, it will be easy to remember the other one. So at least two to three differences you should remember. So this is the answer for question B. Now we'll move to the next question. The next section, that is section number 16, answer the following question. Here also we have to answer only one question which carries 5 marks. So total marks we are going to get 5, and this again has a OR option. So first we'll see the main question which has A and B part. Also it has a C part. So what is the role of placenta at the development stage of the embryo in the female uterus? So what is the role of placenta? We have to explain, which is very important question. Many of the times these questions will be repeated. So placenta, it is the main connection between the fetus and the mother. So the first point, it supply important nutrients like oxygen, or nutrients means glucose and oxygen from the mother to the fetus. So here I'll make some correction. There is a spelling mistake. Sorry for the mistake. So it is Foetus, fetus. So we will move with the next. That is, so it supplies important nutrients like glucose and oxygen from the mother to the fetus, supporting rapid growth and organ development. So whatever nutrients and oxygen required for the fetus for its growth and for organ development, that all will be supplied with the help of placenta. And even it carries metabolic waste and carbon dioxide from the fetal blood back to the mother's circulation for the elimination. So once the fetus takes nutrient and oxygen, after consuming those all, it will produce some waste also, metabolic waste, and that waste will be again sent back to the mother's blood, and through mother's blood it will be circulated and carried to the kidney in the mother, where it will be filtered and eliminated. Even one more important work, that is, it transfer antibodies from mother to the baby, providing immunity against infection. So to prevent from the infection, to be safe from the infection, whatever antibodies required for the baby, those all carried from mother to the baby from this placenta itself. So these are the main important role which placenta will do. So what is placenta? It is a disc-shaped tissue which is the connection between the mother and the blood. So whatever nutrients or oxygen supply required for the fetus to grow organs or to rapid growth, that all supply will be done with the help of placenta. And even whatever waste is produced by the fetus, that all will be carried from the baby to the mother with the help of placenta, with the help of blood. And even supplying antibodies to the baby will be done with this help of placenta. So this is the main organ which help uh, to grow the baby into the mother's womb. So these are the roles of placenta. Now we'll see the question number B. Testes are located outside the abdominal cavity in males. Why? Again, an important question. So testes are the male reproductive organs which are located outside the abdominal cavity, which is called a scrotum, in the scrotum, because sperm production, the testes are the organ which produce sperms, and the process is called as spermatogenesis. Production of sperm is called as spermatogenesis, and this process requires cooler temperature, about 2 to 3 degrees Celsius lower than the core body temperature. So our body has one particular temperature. So compared to our body, the scrotum or the testes require little bit of less temperature, 2 to 3 degree lower temperature, for the production of sperms or for spermatogenesis process. Because at hot temperature, at this high temperature, this spermatogenesis process will not occur, and therefore to perform this function properly and produce viable sperms, active sperms, the testes are located outside the abdominal cavity in the scrotum. So this is the reason why the testes are located outside the abdominal cavity. So this is the answer for question B. Now we will see the answer for question C. So hello students, now we are going to see question number C, that is, how is the number of chromosomes re-established in sexual reproduction? And we have to explain this answer. So here the question is, whenever a new individual takes birth by sexual reproduction, the number of chromosomes will be re-established. Means the number of chromosomes in the young one will be exactly equal to the number of chromosomes present in the parent. So how it is re-established? Because as we know, in sexual reproduction, male gamete from father and female gamete from mother will combine to give a zygote, and that zygote will grow, and then it becomes a new individual, and then new individual will take a birth. So if two gametes are combining, then the number of chromosomes in the young ones, how those chromosomes are established or re-established, that we have to explain. So the answer is, it can be achieved in two steps. So the first step is meiosis, the cell division. There are two types of cell division, that is mitosis and meiosis. So in normal cells, mitosis division takes place, and especially in reproductive cells, meiosis cell division takes place. So here we are studying reproduction, that is sexual reproduction. Therefore, we have to study meiosis. So first we will see what is meiosis. That is nothing but the gamete formation. Specialized cells in the parent organism undergo a type of cell division called meiosis. So there are some specialized cells in the sexual organs. After at a particular period of a life, they will get mature and perform one special type of cell division which is called as meiosis. Meiosis is a reduction division. So meiosis, it is a reduction division. Reduction means what? What is the meaning of reduction? Reduction means reducing. So whatever number of chromosomes will be present in the parent cell, in the main cell, those will be reduced, means decreased into the daughter cells. That is the meaning. So it is a reduction division that results in the formation of gametes like sperms and egg cells, each containing only half the number of chromosomes found in the normal body cells. See, it is very important. So it is a reduction division where what it will do, whenever the gametes are formed like sperms or the egg cells, the number of chromosomes will be half compared to the normal body cell. So here reduction division occurs, and if the normal body cell has 46 chromosomes, that is 23 pairs. 23 pairs means total 46. If it has a 46 chromosome in a normal body cell, then it will divide to give a daughter cell which is having only 23 chromosomes, means half of that. And this half of the chromosomes will take part in the reproduction. So for example, in humans, normal body cell have 46 chromosomes, just now I explained. And meiosis produce sperms and egg cells, each with 23 chromosomes. See, it is very, very important to remember. So here I'll write in human, so I'll write it here male, and here I will write female. It is just to explain you people clearly. So in male, normal body cell has 46 chromosomes. 46 chromosomes. So here in females also it is 46 chromosomes. 46 chromosomes in terms of pair, if we say, then it will be 23 pairs of chromosomes. Here also it will be 23 pairs of chromosomes. So in terms of pairs it is 23, and in terms of total chromosome it is 46. So when cell division, meiosis cell division takes place in case of males, whatever gamete it forms, which is called as sperms, these sperms will have half of the chromosomes compared to the normal body cell. So sperm will have, so here I will draw the sperm. So the sperm will have 23 chromosomes. Remember, very important. So gamete will have 23 chromosomes. Even in females, egg, whatever egg will be produced, so I'll write it here, egg, and then I will draw a egg. So this will have 23 chromosomes. And after the formation of gamete, then these two will fuse to give a zygote. So here first step is completed. So as I told here, it is achieved in two steps. So first here, this whole step, it is called as meiosis. So I'll write it here. It is meiosis. Now the second step we will see. Now the second step, fertilization, nothing but fusion. During fertilization, male gamete, that is sperm, fuses with a female gamete, that is egg, to form a single cell called zygote. So in humans, 23 chromosomes from the sperm, 23 chromosomes from the egg combine to form zygote with again total 46 chromosomes, or in terms of pairs it is again 23. So we will write it here. So here I'll write second step, that is fertilization. So when these two gametes fuse, so I'll write like this, we will draw. Here the fusion takes place and we will get a single cell which is called as zygote. And in this zygote, 23 from male, 23 from female, that is sperm and egg, will combine, and here it becomes again 46 chromosome. So I can write here 46 chromosome, or in terms of pairs, we can write 23 pairs of chromosomes. 23 pairs of chromosomes, and this zygote will divide and grows into a new individual. So these are the two steps. Now if you see, in parents there were 23, see in males 23, in female 23, and now in the young one also there will be 23 pairs of chromosomes. And with the help of these two processes, that is fertilization and meiosis, here the number of chromosomes will be re-established. So this is the answer for question C. After this we will move with the next question. Now we will see the OR option from this question. So this OR option again has A, B and C section. So we will see first A section. How are the reproductive processes in Hydra and Planaria differ? So Hydra and Planaria, in these two animals, we can see the asexual reproduction, and how this asexual reproduction is different in these both we have to explain. So the answer is, first we will write for Hydra. It produces by the method called budding. Means here the whatever reproduction takes place, it will be occurring with the help of budding process. A small outgrowth bud forms on the parent's body. So on Hydra there will be a small bud-like structure growth takes place, and it develops tentacles, and after becoming mature eventually it detaches to become a new independent Hydra. So see here it is very simple. In Hydra we can say budding. Bud means what? It is a small outgrowth which will be formed on the parent's body. That outgrowth will starts growing, and after some time it develops tentacle. When it becomes mature, when it is ready to survive independently, it detaches, it gets separated from the parent and grows as independent Hydra. So this is the budding process which we can see in case of Hydra. With the help of diagram also I can explain this. So like imagine, so this is the Hydra here, and these are the tentacles of this Hydra. Just roughly I am drawing this. So this is the tentacles of Hydra. On this Hydra, there will be a small outgrowth develops like this, and this outgrowth will slowly starts growing. Slowly it's getting bigger like this, and after some days, this also will get the formation of tentacles like this. And when it feels it is independently, it can grow now. It get detached from here and fall down, and then wherever it falls down, there it starts growing as an independent Hydra, and from here it will get cut. So this is the parent, and this is a new individual. This is with the help of budding. Now we will see Planaria. It reproduces by the method called fragmentation. So here the reproduction takes place with the help of process called fragmentation. Fragment means a piece, that is called fragment. The formation of a piece is called as fragmentation. The Planaria splits or pigeon. Pigeon means again breaking. A split means formation of species or fragments into two or more species. So Planaria gets cut into two or more pieces, and each piece develops into a new organism, and this is also called as regeneration process. So here imagine this is a Planaria like this. It has two dot like structure which are called as eyes. So this is a Planaria. This Planaria will get cut into pieces, two or three pieces like this. Then this is the first piece from the head side, this is the middle piece, and here this is the end piece. So here it has got cut into three pieces like this. See here. Now what happens? This first piece starts growing into an new Planaria or individual Planaria. It gives one Planaria. This from here also it starts growing, from here also it starts growing and forms the remaining and develops the remaining part, and this also will become a new Planaria like this. And same way, this third part, it starts growing from the top side, and this also will grow and gives a new Planaria like this. So by this process, we get three Planaria from one parent Planaria. And this is called as fragmentation. Why it is called as fragmentation? Because it has splits into three different pieces. It has fragmented and given three different pieces, so it is called fragmentation. Pigeon means also breaking, and also called as regeneration, because whatever part this portion is missing, that part will regenerate. Now see, it has a head, down part it has to develop. So whatever part is missing in this from the bottom, it starts developing. In the middle part, if you see, the portion from the upper side and portion from the below side both has to be regenerated, both will grow, it becomes a Planaria. And in this piece, if you see it, it has a tail like structure, but upper part you don't have. So it starts regenerating the upper part, and therefore it gives a third planaria. So that's why this process is also called as regeneration process. So this is the answer for question A. Now we will see the next question. Now the next is question B. What are the changes that occur in the flower after fertilization? So in flower, once the fertilization takes place, once the pollen grain comes and fall on the stigma, it develops pollen tube. And that pollen, with the help of pollen tube, the male gamete goes and fuse to the female gamete or the egg inside the ovule. Once the fertilization takes place, what will happen next? That we have to explain. So the answer is, after fertilization in a flower, petals, sepals, stamen, style, and stigma dry up and fall off. See here, petals, sepals, petals will be of colored, sepals will be of green colored. In some plants, sepals also will be colored, but majority times sepals will be of green color. Stamens, nothing but male reproductive part. Style is the female reproductive part, and stigma on which the pollen grain will fall. So these all work will get over. Petals will attract the insect. With the help of those insects, the wings or the legs, pollen grain will fall on the stigma. Sepals will hold the flower. Stamens, male reproductive, style, female reproductive. So these all parts work will get over, and therefore once the fertilization takes place, they all will get dry and fall off. They will all fall off. While the ovule develops into seed. Ovule is nothing but the one where it has that zygote part. So that will convert into seed, and beside all, ovary will get enlarges and ripen into the fruit. So ovary will become a fruit and ovule will. So if you see, the ovary will be like this, and this will be the style, and after the style there will be a stigma. So here I will draw it neatly. So like this, it will be a ovary. So here there will be stigma on which the pollen grain will form. This is the ovary, and there will be many layer like this, and inside this there will be somewhere here ovule. So the pollen grain will fall on this and forms a pollen tube, and from this pollen tube the male gamete will enter inside the ovule. So this ovule which is present inside, that will become a seed. And the portion which you can see outside, this whole portion that is called ovary, this will become a fruit. So this will become a fruit. And this all style, this is a stigma, anthers, all will fall off. And only this much of portion will remain as a fruit. Outside is fruit, inside ovule will be a seed. Now see here next. And the ovary enlarges and ripens into a fruit containing seed, with the zygote inside the ovule forming the embryo. So these are the changes which takes place after fertilization in plants. Now we will see the next one, that is C. Now the next question that is C is, how does a seed grows into future plant? So once the seed formation takes place, once the fruit formation takes place, fruit will be having inside seed, and that fruit will be used to eat by the animals, but the seed will remain as it is, and seed is responsible to give the future plant. So how this seed will grow, that we have to explain. It is there in the NCERT book. Same thing I have, same notes I have taken here. A seed grows into a plant through germination process. See here, germination takes place and seed will grow into a plant. What is germination? Germination is nothing but it is a process where it absorbs water. Absorption of water by the seed is called as imbibition. Remember this process. Imbibition means nothing but absorption of water by the seed, activating enzymes that use stored food for energy. So seed will be having some stored food and some enzymes, and there will be an embryo. Once the seed absorbs water, that absorption of water will activate the enzyme. And with the help of this activation, whatever food is stored inside the seed, that all will be used as energy, causing seed coat to split. So seed will be having a very hard coat. That hard coat will get split by using this food energy. And the roots will develop. It develops two parts actually. One part is called as radicle, which goes downwards inside the soil. That is going to become a root. And the other part which grows upward, it is called as plumule, which is going to become a shoot or the stem, developing into a seedling. And with the help of this process, slowly a seedling will develop, and this only grows and gives a new plant. So if this is a small seed like this, here it will be having a part that is embryo, and these are the two coats. This is one coat and this is the another coat. And this seed, it uses that stored food energy, and these two coats will get open. And once it open, it will become like this. This is one coat and this is another coat. Like this, it will get open, and this embryo will starts growing. How it grows? It grows into two parts. So one part will be like this upside, and the other part will be like this downside. This upside part is called as plumule, and this downside part, it is called as radicle. Remember this. This diagram is there in the NCERT book. Radicle. This plumule will starts growing to give shoot or stem, and this radicle will start growing to give roots. This whole process, it is called as germination. And when this germination takes place, it takes place when the seed absorbs water, that is imbibition. So whenever the seed absorbs water, the process imbibition takes place and it activates the enzyme which is present in the seed. Then it uses the stored food material, it splits the coats which are there on the seed and starts growing into a plumule and the radicle, and it gives a new plant. This is the answer for question C. I hope you have understood all the answers which I have explained for this part C biology of model question paper 2, 2025-26. After this, I'm going to start with the model question paper 3 for the same year, and I'm going to upload all the videos. If you have any doubts or any questions regarding this biology, physics or chemistry of SSLC science, you can comment those all the questions in my videos so that I can answer all those questions and I'll upload the videos. It will help to all the students. So if you like my videos, share my videos with your friends. Comment the doubts which you have in the videos or any other syllabus like physics, chemistry, maths, biology. And please consider subscribing my channel so that I can prepare more videos and upload those all the videos for you people. It will help you. Thank you. Thank you so much.